time to start. So I'm very happy to have David Brückner visit us today. David is an assistant professor of theoretical biophysics at the Biozentrum Basel in Switzerland. And his group studies the physics of living systems with a focus on understanding the principles of embryonic and organ development and the role of single-cell noise in those processes. David started his career as an undergraduate and master's student at the University of Cambridge, UK, and he then got his Ph.D. in physics from Ludwig Maximilian University in Munich, Germany. And after that, he was a postdoc at the Institute of Science and Technology Austria, where he worked with Eduard Arnezo and Gasper Tachik on a range of different projects before he started his group in Basel earlier this year. David has won several awards, including the Gustav Hertz Prize of the German Physical Society and pretty recently an ERC starting grant. And we met in 2023 in Santa Barbara during the quantitative biology summer school there, and we've been collaborating since then on decoding cell signaling using information theory and machine learning. So again, I'm very happy that he's visiting us in person now, and I look forward to his talk.
Yeah, thanks very much for the kind introduction and for inviting me here. I'm excited to tell you a little bit about our recent research on understanding the flow of information in self-organized developmental and multicellular systems. So the fundamental problem that we're interested in is how a single fertilized cell can transform into a fully formed and functional embryo. Now, to undergo this drastic transformation, developmental systems undergo complex multicellular dynamics and ultimately have to make cell fate decisions. So what you see here is kind of a segmented version of a gastrulating mouse embryo, and the different colors are the different cell fates. And essentially, single cells in this tissue have to make decisions on what type of cell they're going to become. Now, they make these decisions based on all kinds of extrinsic inputs, such as signaling from cell to cell or tissue mechanics. And one of the things that makes this decision-making challenging is that when you look at these systems at the cellular and molecular scale, they're intrinsically stochastic. And what this means is when you look at any given single cell in a live recording of such a movie, both its motion but also its chemical state is subject to random and unpredictable fluctuations and heterogeneity. Now, since these cells have to make decisions, what this inevitable presence of noise means is that the amount of information that any single cell has access to make these decisions is limited. Now, the way that the system in which kind of this thinking about cellular decision-making and information theory was pioneered is the early fly embryo, in which there's an externally provided gradient through maternal mRNA that is deposited at one end of this egg. So this is the so-called bicoid protein that diffuses in this embryo. And that establishes a gradient that essentially instructs the cells where they are in the embryo and therefore what decisions they should make and what genes to express. Now, if this gradient was perfect and free of any such noise and fluctuations, then in principle, every single cell in the system could know exactly where it is in this embryo and therefore make a perfect decision with very high accuracy. But in reality, because of these noise and fluctuations, these gradients are stochastic, and that limits the information available to these single cells. And to make that idea intuitive, if this gradient would contain one bit of information about the position of these cells, then this would be sufficient for cells to make an accurate binary decision between two possible cell fates. And similarly, if you had 2 bits of information, that would be enough for patterning precisely into 4 different cell fates. However, the central problem that I want to discuss in this talk is that this positional information framework does not really explain how developmental systems can self-organize. And such self-organization is highlighted in particular by developments in stem cell biology, where we can actually grow patterned multicellular systems, so-called organoids, starting from single stem cells. So for instance, in these intestinal organoids, you can actually start with a single stem cells. Those cells will divide and grow the system, perform morphogenesis, and actually spatially pattern the system. Now in this system, there's no mother that is connected to these cells that somehow tells them or gives them a gradient of where they are in the system and therefore how they should pattern. But it's the cells talking to each other to collectively self-organize this pattern. The other system I discuss in this talk is the system studied by Itz's lab, which is the 2D human gastruloid, where again, the system is not connected to an external source of information, but there are these complex spatial-temporal dynamics of signaling that ultimately lead to a spatial sulfate pattern. So the central question that guides our research is how single cells can obtain information on the decisions that they make in such self-organized systems. The way we often think about these self-organized systems is that there's an initially sort of uniform assembly of identical cells, and these cells talk to each other to then establish spatial inhomogeneities or patterns of cell signaling. And based on these pattern cells, then make these sulfate or gene expression decisions. Now in this talk, I want to look at this kind of conceptual picture here at multiple different levels. The first one will be an experimentally driven part on understanding this initial step of cell-cell interactions that drive inhomogeneity of signaling, where we'll use intestinal organoids as an experimental model. I will then discuss a more theoretical and abstract part where we actually ask the question of how we can define information in such self-organized systems and what that can teach us. And in the final part of the talk, this will be another experimental part about the collaboration with Itze Himskirk on how cells interpret these patterns of cell signaling to make fate decisions in 2D gastruloids. So this first part is about intestinal tissue patterning, and the way the intestine is organized in humans and mice, for instance, is that there are these structures called villi. So these are up here. Here, there's all these different types of cells that are highly specialized to secrete the types of enzymes that are important for the digestion of our food. And because this is a very rough environment, there's constant bowel movement, and it's a low pH, etc. It's kind of a rough environment for a cell to live in. And so they constantly migrate up this villus and actually extrude and die at the top, which means that they need to be replenished. And the only cells that have the power to proliferate in the system are the stem cells that sit at the bottom of these crypts. And one key question is therefore, how during development or also in regeneration, this intricate pattern of these sulfates actually emerges. Now, developments in organoid technologies showed that a lot of this can actually happen through self-organization. So if, again, you start with a single cell and you let the system grow, at some point there's a symmetry breaking event where one or a small number of cells is going to perform the first sulfate decision in the system and differentiate into one of these crypt-like cells, specifically a PANF cell. And then later, this actually leads to a patterning and morphogenesis to generate tissue patterns that look very similar to what you see in vivo. Here, we're going to focus on this very first part to ask whether there might be simple dynamical rules that govern this initial symmetry breaking process. Now, this is a collaboration with the lab of Prisca Liberali, who's also in Basel, and her postdoc, Cornelia Schweier, and PhD student Silvia Barbiero. And what they're doing is to look at some of the key signaling pathways that are important in this process. One of these is DLL1, which is a delta ligand that's involved in Notch delta signaling, and that is expressed by these cells that perform this first sulfate decision, these PANF cells. Another pathway that's important is YAP signaling. And what YAP signaling does in the system is that it sets up initial heterogeneities where some cells are active in YAP, some cells are less active. And when you statistically correlate these two species, what you see is that the delta positive cells are almost always also YAP positive, but not every YAP positive cell is becoming a delta positive cell, suggesting that it's somehow a necessary but not sufficient predictor of symmetry breaking, or of which cell is going to break the symmetry. Furthermore, we also know that if you inhibit YAP, then you no longer get symmetry breaking. But interestingly, if you activate YAP so that all the cells are active in YAP, you also don't get symmetry breaking. So it's really the heterogeneity in the signaling landscape that's important for symmetry breaking to be able to occur. So this is what we wanted to understand from a theoretical perspective. And for that, we were inspired by a long tradition of thinking of these cell-fate decisions in terms of dynamical systems, which was proposed originally by Conrad Waddington already in the 50s. And basically, the way we often think about this is that there are single cells that are subject to external signals. And these external signals modulate the kind of decision landscape of these cells with different attractors in this landscape corresponding to different cell fates. However, in this system, we're not looking at a single cell that is subject to some external signals, but there is a network of cells that are connected to each other. So in principle, each of these cells has its own little dynamical system, and these dynamical systems can potentially couple to each other and also couple to this background tissue heterogeneity. And so what we'd like to understand here is how cell-cell interactions and heterogeneity interplay to determine the symmetry breaking. So to start thinking about this, we started thinking about this delta notch pathway, which is known to engage in a process called lateral inhibition, where essentially every cell tries to produce this protein delta, and that expression of delta, which is bound to the membrane and binds to notch receptors of the neighboring cells, inhibits the production of delta in the neighboring cells. And so on a 1D arrangement of cells, that essentially leads to alternating on and off states of delta signaling. Now you can describe that system using a very simple chemical kinetics equations, where basically the production of delta, as denoted as D here, is inhibited by the lateral inhibition signal that any given cell receives, which is simply the sum of the delta of the neighboring cells. Now you can simulate the simple model on a spherical arrangement, since here we're not dealing with a 1D line of cell, but this kind of spherical organoid. And we're doing that at around the 16 cell stage, because this is where the symmetry breaking occurs in the experiment. So the spherical topology then determines the connectivity network of these cells. And then actually, this model, of course, predicts symmetry breaking, but it then also makes a parameter-free prediction of how many of these cells are going to become delta positive, which only depends on the connectivity of this network. Specifically, what this model predicts is that around a quarter of these cells should become delta positive, which is actually significantly higher than what we see in the experiment, where it's around 2 out of 16 or 1 in 8. So this means that we really have to consider additional factors beyond just standard lateral inhibition signaling in the system. And so we started thinking about how we could connect lateral inhibition signaling to this tissue heterogeneity of YAP. And to do that, we wrote down this more general dynamical system, where now the delta state of every cell is not just a function of the lateral inhibition signal, but can also be modulated by the YAP level. What we then did was to scan through possible combination of activation and inhibition functions to that essentially make up this dynamical system. And we tried to look for the simplest combination of these functions that would capture the qualitative observation that if you either inhibit or activate YAP, you no longer get symmetry break. When we did that, the simplest candidate that emerged was this combination of 3 functions. So first of all, we have here a baseline activation of delta by YAP. Then there's the inhibition function as a function of the lateral inhibition signal, as expected. And then actually there's an additional auto-activation through delta that will become relevant in a minute. This auto-activation actually leads to this kind of non-trivial shape of this 3D manifold that now essentially determines what states are available to these cells as a function of the lateral inhibition signal that they receive and their cell intrinsic YAP state. So to make this a little bit more interpretable, if we focus on a cell that is low in YAP, what this system does is to make the response of these cells to the lateral inhibition signal a little bit more complex than you have in the standard lateral inhibition model. Normally, when you receive low lateral inhibition signal, you would expect to just be delta positive. But here there's a cell intrinsic bistability where actually cells can still be in a stable delta negative state when they receive low lateral signal, which is separated from the high delta state through an activation barrier. On the other hand, the YAP high cells, they have this standard lateral inhibition curve, but here with this offset that will become relevant in a second. So what this model does is it essentially takes this random input heterogeneity and feeds it through this dynamical system, coupling interactions and YAP signaling. And then essentially the cells that are low in YAP because of this bistability are essentially disqualified from this lateral inhibition process. Some of the YAP high cells can become high in delta, but not every YAP high cell can do that because some of these YAP high cells actually neighbor another YAP high cell and are therefore sort of laterally inhibited by that cell, and only one of the two can become delta positive. Now, why does that explain this non-trivial YAP activation experiment? Essentially, what our model here is saying is that because YAP is providing a baseline activation of delta, all of these YAP positive cells have an intermediate delta state, which is what you see here, and that is just sufficient for all of these cells to collectively block each other from becoming properly delta positive. Now, there's a key question here, which is, we've now constrained this three-dimensional landscape based on these perturbation experiments, but does this landscape actually make sense molecularly or mechanistically as well? And one of the things that we realized here was that in order to get this bistability, we need this auto-activation loop on delta. And this is what our collaborators told us is actually a little bit weird, molecularly speaking, because delta is a membrane-bound protein, and it doesn't have any known signaling pathway back into the cell nucleus to modulate gene expression. So what we then did was to essentially try to write down a slightly more complicated model where we introduced an additional species that we just called X for the moment, which couples YAP to delta and can perform autoactivation on itself. So this could be some transcription factor, for instance. Now, if you take this model and you again kind of derive this three-dimensional landscape here of delta as a function of lateral signal and YAP, you still get exactly the same landscape. So it's basically a slightly more complex mechanistic model, but it has exactly the same mathematical structure as the model that we inferred from the data. Now, of course, it raises the question of, what is this intermediate variable X? So for this, in Priska's lab, they then followed an omics approach where they used single-cell ATAC sequencing to find transcription factor binding motifs that have increased accessibility in the cells that are on their way of becoming delta positive. And what they found was that there was one transcription factor in particular, FOXA1, that had a strong enrichment of these kind of binding motifs, leading to the hypothesis that maybe this X factor could be FOXA1, which is something that now, of course, then we had to test from the experiment. Now, interestingly, our model, our extended model with this X factor actually makes predictions for what we would expect this intermediate variable FOXA1 to do in terms of protein localization in all of these experiments. So first of all, it predicts that in the control experiment, FOXA1 should be high in the delta positive cell, which is indeed what we see in the experiment, and that it should essentially not do anything in either the YAP inhibition or YAP activation experiments where we do not get symmetry break. However, we then also wanted to use our model to actually make new predictions for perturbation experiments that we did not use to constrain our model. And the first one of these was a so-called DAPT treatment, where you essentially get rid of the ability of these cells to laterally inhibit each other. And then what you very generically expect is that you see many more delta positive cells. Now, our model predicted that these delta positive cells should then also be FOXA1 positive, which is indeed what we saw. But then we came up with two additional perturbation experiments that I think really provide a non-trivial test of this model. The first one was where we now both get rid of, where we both inhibit YAP and we get rid of this ability of the cells to talk to each other. And then again, normally when you treat such a system with DAPT, you would expect to see many more delta positive cells. But our model is saying that because the cells are low in YAP, there's a cell intrinsic bistability, meaning that they would still remain in this delta negative state, and therefore there wouldn't be symmetry breaking. And that's indeed exactly what happened also in these experiments. The next experiment was one where we combine this DAPT treatment with the YAP activation. And here, just to remind you, with the YAP activation experiment, essentially what we were saying was that the reason why you do not get symmetry breaking is because all of the cells have an intermediate delta level and are blocking each other from becoming delta positive through lateral inhibition. So now if we get rid of this ability of the cells to laterally inhibit each other, then all of the cells should become delta positive. And this is actually exactly what happened in these experiments as well. So with that, I would like to wrap up the first part of this talk. So essentially, what we're saying here is that there are simple dynamical rules that essentially take this initial noise and heterogeneity, pass it through this dynamical system, and then return a discretized sulfate pattern that is consistent with what we see in these experiments. With that, I want to also come back to the overview here. So we've discussed now this first part on intestinal organoid self-organization. And I'd now like to start the second part on a slightly more theoretical question, which is how these systems where cells are communicating with each other, like in these intestinal organoids, how we can define the amount of information that has been self-organized in the system. And to start discussing that, I want to contrast these self-organized systems like intestinal organoids with a different type of development that we're more used to thinking about, which is instructed development, which happens, for instance, as mentioned in the introduction in this early fly embryo, where essentially you have this extrinsic input that provides information to the cells on what kind of decisions to make. Now, there's quite a large body of theoretical work looking at these kind of instructed developmental systems from an information transmission perspective. And the way that we can think about this is that the system is fed this input signal, which is passed through a channel, which is essentially every single cell taking the input and computing the outputs, and then provides an output pattern. Now, if you want to know, given the input signal, what would be the optimal channel in order to perform this process, then you can look at an information theoretic object, which is basically the amount of information that has been transmitted from the input to the output, which is the mutual information between the two. So basically, we would like to maximize how much information the output carries about the input. However, this is really fundamentally different to the problem that we're thinking about in self-organization, because here we have this interacting network of cells that is just subject to initial noise and heterogeneity and does not really receive any input signals, but then returns an output pattern. So the question is here whether for this type of system, we can also define the information in this output pattern and perhaps use that as a way to measure the performance of these systems. So to start thinking about this, we can kind of use this kind of cartoon embryo here, where we just have a couple of cells that are talking to each other in a one-dimensional arrangement and make these decisions into these discrete types of cells that are shown as the colors here. Now, because this process is stochastic, we're not going to end up with a single deterministic pattern, but actually a distribution of our patterns. And the way I'm going to plot these distributions is as this 2D matrix here, where basically here is the one-dimensional embryo axis, and then we have multiple runs of the system or multiple copies of these embryos on the y-axis. So essentially what we want to achieve with this performance function is to distinguish systems where nothing interesting at all or something very random happens every time that you run it, from systems where either we achieve perfect positional order, or there might be non-positional but clearly very non-trivial kind of correlated patterns emerging. And the measure that we came up with is this function here, which is a so-called Kohlberg-Leiber divergence, which basically measures how different two probability distributions are. And the way we're defining this is that we are comparing the observed distribution of sulfate patterns to a maximum entropy distribution generated from these patterns. And a simple way to imagine this is that the one on the left is simply you take all your embryos or organoids, you dissociate all the cells, throw them in a bucket, and then randomly reassemble your tissue, okay? And what we're essentially measuring here is how much additional information is contained in the real observed patterns due to the presence of, spatial order. Now that function actually also decomposes into two interpretable terms. The first one is the so-called positional information, which measures, again, the mutual information between the cell fate, so the color and the position, and is therefore maximized by a positional pattern like this. And the second one we call the correlational information, which measures the information in this type of pattern here. So here, there's actually no positional information because at every location, you're equally likely to find a red, green, or blue cell. But clearly, it's not a random pattern, right? And the reason why it's not random is because the pattern is spatially correlated. And the amount of information contained in these spatial correlations is exactly what this function here measures. So what this approach suggests is that we can decompose the self-organized information in these systems into positional and correlational information, and we can therefore draw up this two-dimensional phase diagram of possible outcome patterns here. So basically these random patterns, they're down here with zero information. Then positional patterns have high positional information, and the correlational patterns are somewhere up there. But then there are, of course, a lot of patterns that you could imagine that are somewhere in between in this 2D space. Now, this might all seem, like kind of mathematical playing around. It's kind of fun, but, what could we actually do with that? And I want to suggest two things that we could achieve with that. The first one is that this idea provides a possible optimization principle for signaling networks. So if we now think about, all the parameters that determine the system, we could maybe find optimal ones. And the second one is that we actually would like to estimate such information directly from high-resolution experimental data. So in the first possible application here, these signaling networks that allow these cells to talk to each other, they are, parameterized by many parameters, like all the reaction rates, the diffusion constants, et cetera, et cetera. And in the previous part of the talk, we basically asked, given some particular system, we have some data on the system, can we figure out where in this parameter space we need to be in order for our model to predict our experiment? But you can also imagine asking a more general question, which is where in this parameter space is self-organization possible or robust in the first place? So I want to give you a very minimal example of how we can apply that type of thinking to a very simple system. But then also, if you're interested, you can check out this paper where we applied this to all kinds of other things like Turing patterning and so on. So this again here is this kind of lateral inhibition signaling where I've now introduced a single parameter alpha that essentially tunes the system between a monotonic response of, so the response function of the cells is a function of the input signal that they get, and a second term, which is the self-activation of the cells. And if this alpha value goes beyond some critical value, then you actually get a bistability, kind of similar to what we saw in these intestinal organoids. What's interesting about this is that if you imagine that your system is completely free of noise and fluctuations, then it actually doesn't matter what value this alpha has, because wherever you are in this alpha parameter space, you always get these alternating patterns of on and off cells. But now if we slowly ramp up the intrinsic noise magnitude in the system, which is basically the intrinsic gene expression noise, then there's actually an interesting optimum. So first of all, if your noise is low, it's actually better to be in this monostable regime, because then your curve kind of gives the cells an accurate response based on the signaling that they receive. And you avoid all kinds of problems of being stuck in these attractors from the bistable regime. Whereas if the noise is large, then actually you don't want to be sensitive to an extremely noisy signal, but it's actually better to be bistable and to have these kind of cell-fate attractors, like in the Waddington's landscape, that the cells can kind of hold on to in the face of noise. So that regime is actually kind of reminiscent of what we saw in these intestinal organoids, suggesting that cell intrinsic bistabilities can be both important in sensing heterogeneity, but also to buffer against intrinsic noise. Another interesting perspective, I think, is that in these parameter spaces, there might be large regions where it's possible to perform self-organization in a deterministic setting. But when we actually consider the inevitable presence of stochastic noise in these systems, then that can actually shape the optima and therefore constrain the regulatory mechanisms that these signaling networks undergo, such as things like autoactivation. Now, the second possible application of this framework that I want to highlight here, and this maybe gets back a little bit more into the interest realm also of the spatial single cell analysis program, is to actually try and measure this directly based on single cell spatial experimental data sets. And this is where I transition to the second experimental part on 2D gastruloids. Now, as I mentioned, this is a collaboration with Itze Himskirk's lab and his students, Basel and Seth. And what they're looking at is this 2D gastruloid system, which is basically this flat pancake of cells where, sort of at the beginning, you have identical human pluripotent stem cells. And then there's a medium that sits on top of these cells that contains some of the key morphogens in the system, one of which is BMP. Now, if you look at the system again around 2 days later, then what you see is this radially ordered cell fate pattern here. And at the same time, you also observe that there are spatial patterns of some of the key signaling pathways that we know to be important in these early developmental processes, including BMP, Wnt, NOLA, and FGF. So some of the key questions that we can ask here is, first of all, how precisely do these signals define positional information in the system? And does that information actually explain spatial sulfate patterns at the single cell level? So the data set that we're working on here is this iterative immunofluorescence data set, where in the same colony, and therefore in every single cell, we have a measurement of both the signaling state of these cells, so these kind of downstream effectors, like, a phosphorylation of PSMAD1 tells us about the local BMP state, and then also the sulfate markers, which are the genes that are expressed by these cells. Now, importantly, this method is very quantitative, because if you, in these iterative round of staining these different pathways, you actually stain the same gene multiple times, you see a very strong correlation with the technical noise that is much smaller than the biological scatter, meaning that we're actually measuring the biological noise in the system when we're looking at probability distributions. So based on this data set, the question that we started asking was whether, based on these spatial cell signaling, we can actually predict the gene expression patterning, or in other words, how much information do these signals contain about the gene expression? Now, importantly, when you look at these signaling patterns, they actually have quite different spatial distributions to some of these genes, and therefore it's not obvious sort of what combination of these signals would tell us, whether a cell is nanopositive or negative. So to address that question, which is essentially a complex high-dimensional nonlinear regression problem, we started using simple feed-forward neural networks, where we're feeding in at the single cell level the measured signals to predict the gene expression outcome. Now, strikingly, when we applied this approach, we actually found that this works extremely well. So first of all, we capture the spatial patterns of the gene expression that you see here. So here, these are the radial profiles as a function of the distance from the colony edge. And then also, if you classify these cells into either positive or negative cells for each gene, if you look across these genes, we get an average accuracy of positive, negative predictions of around 90% at the single cell level. So this means that these signals actually contain a lot of information about this gene expression. But of course, it also raises the question of whether this is somehow, a very specific type of correlation that we see here in this experimental condition, or whether this is actually a generalizable, biologically meaningful map. So to test that, essentially, what we did so far essentially was to train this model on the standard condition here, which is defined by a particular concentration of the BMP4 that's initially in the medium surrounding these stem cells, which is around 50 nanograms per milliliter. And that is a parameter that you can vary. And when you vary this concentration, of course, as you get more and more BMP, you actually get more and more PSMAD, which is downstream of BMP, and you get this longer range gradient. But interestingly, because the system is in constant crosstalk, and these other signals are actually downstream of BMP, all of the other signaling patterns are also changing quite dramatically. So for instance, if you look at LEF1, which is downstream of Wind, then you have much less of that in B10, and this ring kind of has gone radially inwards in the B200 system. So the question is now, if we use our model that was trained only on this B50 condition, can we actually predict how the sulfate changes when we look at these other conditions? So to do that, I will represent here the gene expression outcome as a discretized or thresholded version, where essentially based on all of these genes that we measure, we classify the cells according to interpretable sulfates like the amnion, which is the outermost sulfate here. And what the model now predicts, based on the signaling signatures of this B10 and B200 condition, is that compared to B50, we would see much more amnion, a much broader amnion ring, and in B10 we should see much less amnion. And all the other sulfates, as you see, also kind of redistribute in the system. So this is just what the model predicts. But then if you actually measure, or you look at the gene expression in the system and you do exactly the same classification, then you see that the model really quantitatively captures the redistribution of these different sulfates, and this also holds up at a quantitative level. So this really means that we have this predictive and generalizable signal to fate map in the system, but it also raises the question, how we connect this to this positional information framework that I introduced earlier, where we're now interested in the spatial precision of these patterns. And the first question that we asked here was, given that we see a certain spatial precision in these signaling profiles, does that actually somehow relate to or constrain the spatial precision or positional information in the gene expression profiles? So to address that question, we can actually measure this positional information in the signaling, and you can do that for a single signal. So say if we just look at the SMAD1 gradient, we get around 1 bit of positional information. So that would be enough in principle to make a reliable decision between two sulfates. But now, as you include more and more of these signals, and you add them into this mix to predict position, then you see that the positional information gradually goes up. There is some overlap between these information quantities, so the information is slightly redundant, but it is combinatorial. And if we add all of these signals, we end up with around 1.7 bits. So then you can apply the exact same analysis also to the gene expression profiles. And what we see here is that we end up with around 1.86 bits. So it's a little bit higher than what we see in the signaling, but it's also not super different. So somehow these two patterns, the signaling and the gene expression, have roughly similar spatial precision. Now, this is actually really ongoing work, but there's one more point that I want to make regarding the spatial precision, which is that if we really assume that we can predict the signaling we can predict the gene expression from the signaling, then we would expect that if we have these, if we have certain defects in the signaling, which you see, for instance, here in this colony, so this sometimes happens in this B10 condition, that you have these kind of long-range defects where the SMAD2 gradient, which is downstream of nodal, actually has a shorter decay length over here in the system than over here. So here I've basically unwrapped this profile onto the polar angle around this 2D colony. So our model then predicts, if we have this defect in the signaling, then we should get a very similar defect also in the sulfate. And so we should get this kind of defect here. And then when you actually look at the gene expression pattern, so for instance, the nanonc here, and then also this thresholded, then this is exactly the defect that we see also in the observed gene expression. So this really highlights this fact that signaling and gene expression are extremely highly correlated in the system, and that we can predict one from the other. So just to give you a little outlook where we want to go with this from a theoretical perspective, is to go all the way from the way these cells are interacting and the way that the signaling network is parameterized to predict the spatiotemporal dynamics of signaling in a reaction diffusion model of the system, to then learn how these dynamical signaling trajectories eventually link to the sulfate, which is something that we're starting here with these neural networks, but we're also hoping to do that with really kind of interpretable biophysical models. Now with that, I'd like to wrap up and kind of, again, highlight this hypothesis that in these systems, there's information in these patterns that self-organizes that we can decompose into positional and correlational information. And just to place the two systems conceptually that we've discussed in this talk into this 2D diagram, I would say these intestinal organoids are a nice example of a system that has correlational information, because there is a single cell that has differentiated. It doesn't matter where the cell is. It just matters that we have the right proportion, the right relative locations of these cells. On the other hand, in the 2D gastruloids, the symmetry is actually broken from the beginning because, you know, we have boundary and we have bulk cells. And so there's positional information along this radial coordinate that we observe in these gene expression patterns. With that, I would like to thank all the members of the group and also the collaborators, especially Prisca Liberali and Cornelia Schweier and Silvia Barbiero, who worked on the intestinal organoid problem, and then Itze Himskirk, Bassett, and Seth, who worked with us on the 2D gastruloids. And with that, I would be happy to take any questions.
So I guess I'm not quite sure how to formulate this question, but in the second part, if I understand correctly, you're looking at immunostained images. So you're looking at signaling output and sulfate at the same time.
Yes.
Right. But you're talking about it as if the signaling is causative of the sulfate. And that would really require that you look at signaling somewhere upstream of the final steady state you're in. So how do you reconcile these things, or are you even thinking the signaling is causative of something?
Yes, yeah. So that's a great question. So you're absolutely right. We're looking at the staining at the same time point. So fundamentally, you know, based on this whole nonlinear regression approach, all we can say, essentially mathematically, all the statements that we can make are about correlation between signaling and sulfate. You could, for instance, also imagine actually training the model to use the genes to predict the signals, right? And that actually works equally well. So, you know, the mapping works both ways. Biologically, of course, we know that signals are somewhere upstream of sulfate, but we also know that sulfate can feed back onto the signal, right? So there can be some kind of loop like that. So one thing that we're doing now, which is really ongoing work, so I haven't shown the results here, is that we actually also perform perturbations on these signals. So you can, for instance, inhibit or activate individual pathways. Then again, we can measure all of these signaling signatures and predict the gene expression. But we can also essentially ab initio try to say, you know, if in the wild type signaling things, we just get rid of YAP or we get rid of ERK, what will we predict? It's still essentially a statement about this correlation, right? So essentially, I think our claim here is that there's a very strong relationship between signaling and sulfate in the system, and that perhaps actually also cells, as they differentiate, they maybe reinforce or feed back onto the signaling signature that induces them. Yeah.
So can I follow up? You could treat signaling outputs and fate markers on the same footing and say that you're just sampling a subset of all state variables you have, and you could pick other random subsets to sample and see which ones are most predictive or most predictive of the unknown ones. Are the signals in any way special if you do something like that?
Yeah, that is a great question. So you could absolutely do that. We haven't done it in that way. So you could look at, you know, if I want to take the minimum predictive set of variables to predict all the others. We should actually try this. I'm pretty sure you will actually pick out things like PSMAD1 and ERK because they really have like very informative spatial patterns, more so than some of the genes. Also, just to highlight, like there is a biological distinction between the signaling readouts and the genes, because the signaling readouts are not gene expression, but it's actually, you know, how much ERK has been phosphorylated or something. So there is a biological distinction there. But I totally get your point. So I agree. Yeah. Oh.
So I was just curious. So for example, in the last experiment that he's asking you about, does it concern you at all that all of the data that you have was necessary and all of the data that you obtained was predicted? So would it not be likely that only some subsets of the data that you would have would predict some subsets of the, do you see what I'm saying? It
Right,
seems
yeah.
weirdly surprising that you have everything is all predicted and you needed all the data to do it.
So say in these different BMP conditions, right? There, at least the model training was only done on one of these conditions, right? So once we have measured that one condition, which was this B50 condition,
Yeah.
then our model is fully constrained, right?
Yeah.
And then we can test the model on the other experiments. But you're right that we are still applying the model to the measured signaling patterns, right?
Yeah.
And we would.
There's nobody that breaks your rules, right? There's no signaling molecules that you said are actually useless, pointless, and maybe counterproductive. And there's no developmental programs that were outside the realm of what you could predict. It all is sort of self-enforcing, if you use all this data to protect all that data, there's no new information coming in, right? You don't say to yourself, oh, well, let me get another developmental program that I didn't train on, for example, and see if I can predict that.
Right, yeah, so I mean, I think that would be, you know, a very ambitious next step that we actually do want to follow, right? So sort of in the outlook here, what we want to do is to actually not just go from measured signals to predicted genes, but to go from, you know, a hypothesized signaling network that we can either learn from the data, but we can also change something about it, right? So we could say, for instance, you know, in the model, say we get rid of this loop here, or we modulate the diffusion of nodal or whatever, how would the signaling patterns change? And then also, what does that predict for the output sulfate patterns, right? And so that would really be an approach where we start all the way, you know, based on sort of mechanistically interpretable parameters to predict the outcome. And then hopefully, of course, that would also be somehow generalizable to other systems, right? So we're actually trying to connect that to some experiments in zebrafish or some things that IDS is also trying on the chick embryo, for instance.
So did you try using, for example, a minimal set of data to drive your predictions instead of every piece of data that he gave you?
So if you mean so we had these different signaling pathways here, right? So we used, in this prediction, we used all 7 of these.
Right.
So you can actually ask the question, you know, if I basically sample all possible combinations of these, right, what would be the minimal set that still predicts and is generalizable? We actually found that this PAKT was not very important for the predictive power. So essentially we could remove it because it's, you know, the information that it carries is essentially entirely redundant with some of the other signals. And then when you look at the other signals, the answer of which signal is important actually changes depending on which gene you're looking at. So for instance, if you want to predict ISL1, which was one of these amnion genes that's expressed really at the boundary, all that you need is PSMAD1 and ERK, and then you can perfectly predict them. Now, there are other genes like NANOG. Maybe if I go back to this slide here, so NANOG and SOX2, they have these very broad kind of expression domains. And for those guys, we actually needed all of the signals, except this AKT. And so, you know, it's really showing that the signaling code is not, you know, it depends on the gene that you're looking at. And it actually tells us something about which signals are important for the prediction of which of the genes. Yeah. And there was another question here.
Thank you for the wonderful talk. I was just wondering about along the same lines of, using your spatial organization of these signals, how important it is and how do you make sure that is robust? Like if you perturb your spatial signals from any of those molecules, like, YAP or something, how, because there could be a lot of noise in the data coming from experiments. This could be very clean experiments, but some other experiments could be very, noisy actually.
Right.
But how does it affect your predictability? And how important is the data to be very robust?
Great. Yeah, that's a great question. So first of all, the prediction here we really did at the single cell level, right? So in every single cell, we look at all the signals and then predict all the genes. So in theory, you can do that without any knowledge or reference to space in principle. However, what was I going to say? Again, when we think about this outlook of actually predicting the signaling spatiotemporal patterns from these signaling networks, we can then also ask, are these signaling networks somehow wired to make these patterns spatially precise, right? So that's something that we're definitely planning to look at.
Thank you. Along those lines, I just have one last question. If you have, I don't, similar to what other person was asking actually, if you have to identify what are my drivers versus passengers, so that you can minimize the input that goes into your model and still you'll be able to predict.
Right.
And also how it is very system dependent, or can you take this knowledge and apply to some other pathway, what do you learn from here, how applicable it is to other signaling network?
Right, yes. Yeah, so we do expect that some of the relationships that we find here will be generalizable, at least to other developmental systems where, the same morphogens are in action, and often the same morphogens induce the same genes and therefore cell types. But it would be something that I think will be really exciting to actually test in other systems as well. Yes. Yeah.
Thank you.
And that was
Have you tried your system and your modeling and formula for like motility or pH changes?
No, we have not. I think that would definitely be interesting to now also start varying a number of other parameters. You could also imagine somehow modulating the metabolism in these systems, I don't know, like the amount of nutrient or something, whether, some of that somehow modulates this landscape of how signals and sulfate are related.
Yeah, great talk. So, the first person, so you mentioned the YAP expression. So that means that the cells may sense now the mechanical environment. Right. So
right.
Yeah.
Yes.
So how are you going to incorporate this data for your model?
Yeah. Yeah. So actually, one of the simplest things that we've thought about doing was, basically from these data alone, we can already infer something like the local cell density, right? Which is actually not uniform in this system. So the amnion cells are less dense than some of the other tissues. And which might also be related to their YAP activity, right? Because the cells are more stretched and therefore they're more active in YAP. And well, it would be interesting, once we can really measure also some of the mechanical parameters to relate that either to predict the signaling or maybe also to add it as a signal into the prediction of sulfate. But again, maybe also coming back to the point about correlation versus causation, right? It's something where we might want to look also at sort of dynamical data to understand perturbations, to understand what is really causative versus downstream of sulfate.
Any more questions?
Nothing on Zoom?
I did not see anything show up on Zoom, so.
Okay, great.
I thought it was a fabulous talk. Thank you so much.
Thank you.